Welcome to the video lesson on meiosis. In this video, we'll be discussing what meiosis is, the different stages of meiosis, and how the end results of meiosis are different in males and females. Let's start with what is meiosis. Meiosis is the division used only for making sex cells, and in biology, those sex cells are called gametes, and they would be the egg and sperm. They result in cells that are haploid. This is opposite of what we learned during mitosis, where the cells were diploid. Haploid contain only one set of genetic information. In humans, that would be 23 chromosomes. Not 23 pairs for 46 chromosomes, but 23 actual chromosomes. Meiosis takes place in two types of stages. We call them Meiosis I and we call them Meiosis II. You're going to recognize the names of these phases because they're very similar in name to the phases of mitosis: prophase, metaphase, anaphase, and telophase. However, because it takes place in two different stages of meiosis, we call them prophase 1, metaphase 1, anaphase 1, telophase 1, and then the same phases again, except a 2 at the end. You're also going to recognize, and thankfully so, that many similar things happen during meiosis as do during mitosis, but just with a few little tweaks. So we'll highlight what those are. It will be a nice review of mitosis from actually what happens during each phase. And we'll also highlight what are the unique things that happen here during meiosis. But again, this is done with haploid cells, not with diploid cells, only for making sex cells. All right, here we go. Prophase 1. Okay. Prophase 1, we have the centrioles split and form spindles. Our nuclear membrane dissolves, and our chromatin coils up to form chromosomes. Now, all this should sound very familiar to you from mitosis. However, in meiosis, tetrads are formed, and tetrads are chromosomes that form homologous pairs. So the chromosomes pair up with each other, and in this process is called synapsis. And here's the other really important thing that happens during prophase 1. It's called crossing over. Crossing over begins in prophase 1. In crossing over, the chromatids from the homologous chromosomes cross over one another. Then alleles from the cross sections are exchanged. Crossing over produces new combinations of alleles on each chromatid. And that's highlighted from page 276 of your textbook. Crossing over is extremely important, and that's how we get variation in humans, by that exchange of genetic material between the chromosomes themselves. This is why you may look similar to a sibling, but not identical to a sibling, and not identical to your parents. All right, next we have metaphase 1. Metaphase 1, chromosome pairs line up down the middle. That should sound familiar. And crossing over continues. They continue to exchange that genetic material between the homologous pairs of chromosomes called tetrads. Anaphase 1, the chromosome pairs, the tetrads, are pulled apart, and this is called disjunction. Telophase or telophase 1, the cell splits into two new cells with one chromosome from each tetrad. So what do we have as a result of meiosis 1? We have two new cells with one chromosome from each tetrad. That's the end result, the end product of meiosis I. Now, each of these cells is now going to undergo a process called meiosis II. And it's going to go through the same types of stages, except now they're called prophase 2, and etc. They just have a 2 after the end. So the same exact process as prophase 1, but no tetrads. The centrioles are going to split and form spindles, the nuclear membrane is going to dissolve, and the chromatin coils up to form the chromosomes. Except this time, they are not tetrads anymore, they are just single chromosomes. They're no longer homologous pairs. Metaphase 2, same process as metaphase 1, but again it's just done with the chromosomes, not with the homologous pair tetrads. Chromosomes pairs line up down the middle. Anaphase 2, the chromosomes split into chromatids. Now this is different than anaphase 1, where the homologous pairs split into single chromosomes. Now those chromosomes split into chromatids. And telophase 2, some nice diagrams here to go with this. New cells form with 2 chromatids each. Okay, so each of them has a chromatid from the split chromosome. Well, let's do the math here. If each cell now splits into 2, what do we end up with? And of course, we end up with 4 new cells, 4 new haploid cells altogether. Now, the result, however, is different depending on if you're a male or a female. In males, this process is called spermatogenesis. This is how males make sperm cells. And in females, this is called oogenesis, how females make egg cells. Now, the process is very similar. You start out with a diploid cell that undergoes meiosis I. You end up with haploid chromosomes. And in the diagrams, meiosis I takes place right here. Okay? What do you end up with? 2 cells each with single chromosomes. And remember, these are tetrads here that have divided into single chromosomes for these 2, okay, and these 2. Then those 2 cells each undergo meiosis II at this point right here, and you end up with 4 sperm cells with 23 single haploid chromatids in each. Now, for spermatogenesis, we end up with 4 sex cells. However, with oogenesis, we end up with 1 egg cell and then 3 polar bodies that result out of this. And these 3 polar bodies end up dying. In animal cells. In plant cells, they don't die. They just are kind of non-functioning except to provide the egg cell food. Okay? And so that's how we end up with sperm cells and egg cells. Alright, next we're going to take a look at a neat video that does a nice job explaining this whole process and also putting it in animation so that hopefully it makes a little bit more sense to you.